My name is Karla Williams, and I'm an assistant professor here in Vancouver at the University of British Columbia in the Faculty of Pharmaceutical Sciences. And one of the research areas that my lab focuses on is on extracellular vesicles. And we're interested in extracellular vesicles for numerous reasons. We're interested in their biology, we're interested in how they interact with other cells, and then what brings me here today is we're also interested in using them as a window into the body, to be able to look at an individual and to be able to determine their health status or their disease status. And so for this, we have been doing a lot of nanoscale flow cytometry. And we've been working with Beckman on their platform, the Saito Flex, to try and develop this technology to a standard where we're happy with what we're seeing. So what I'm going to be talking about today is a couple things. First, just a little bit of background on why are we interested in doing this type of technology? What's some of the clinical questions that we're trying to answer here? And then move on into some more of the technical aspects of it. How does the machine run? How do we look at things on there? And then once we've set things up, how does it look when we're actually trying to develop some type of a diagnostic test that we might see being able to have some type of applicability within a clinical setting? So to begin, we'll start with just looking at why would it be advantageous to add some more clinical tests into the clinic that could improve how we detect cancer. And some of the notable cases here would be in our looking at our detection rates for prostate and breast cancer. These are the two most heavily diagnosed cancers. However, when we look at the incidence of mortality associated with these, those levels don't quite correlate. And while that's in part because we do a good job of treating these cancers, it's also in part because there's a significant amount of overdiagnosis in these types of cancers. And this is something where putting in a new type of a testing platform to try and do better risk stratification could be really beneficial. Now we have another issue when we talk about cancer diagnostics, is we can also see that there's a few cancers that show up on this list, such as pancreatic and ovarian, that they're not represented as some of the top cancers that are diagnosed, but yet we see that they cause a significant amount of mortality. And that's because we lack diagnostic tests for these. And so what a lot of times happens is for these types of cancers, you have patients presenting, the majority of them presenting in late-stage cancers. These late-stage cancers are not treated very well. You can see that here for pancreatic, and then you can also see that here for ovarian cancer, where if you can catch it early, the survival rate is quite impressive. But most of these patients, unfortunately, present at a later stage. So all of this really highlights the fact that we need improved methods to detect certain cancers, and then we need improved methods to risk stratification within these cancers. And so this then really brings us to the world of liquid biopsies. So what we want to do is we want to be able to find out information about how somebody's body is functioning without having to do elaborate scanning. Because we know that's one of the things that we have is for some cancers that we have, such as breast cancer, the scans that we can do, which are mammography, they often will show small lesions that are indolent, and that's where we get our overdiagnosis problem from. And similar for prostate too is we pick up a lot of small indolent cancers in prostate. So what we'd like to do is we'd like to be able to take blood samples and we'd like to be able to look in the blood and see what type of information can we get. Because if you look within here, here's our cancer cells within the tissue, and we can see all sorts of different components are released from the cancer cells. And being able to tell which ones are cancer-related and which ones are healthy can kind of give you a bit of a window into whether or not an individual has a disease state. And there's been some good examples of these in the field across different platforms. We can proteins and metabolites is one where we have some liquid biopsies that have been developed by just looking at the proteins and metabolites that you can find in someone's blood. A notable example of this is actually the PSA testing, so the prostate-specific antigen testing. It does cause overdiagnosis, but when it came into the clinic in the 80s, it was a great way to be able to catch prostate cancer early. We're now looking to try and better this to do some risk stratification, but I will get into that later on. Now the other thing is nucleic acids. So we find a lot of nucleic acids, a lot of cell-free DNA and circulating tumor DNA. And so there's been some good progress made in these, especially in terms of lung cancer and looking at EGFR mutations. And now another platform for liquid biopsy development is circulating tumor cells. And this area has had some difficulties in terms of looking at marker composition on flow cytometry and having low event numbers. It can be useful in certain types of metastatic cancers, such as prostate and breast metastatic cancers seem to have elevated levels of CTCs, but there are some issues associated with these types of technologies. Now we look at extracellular vesicles. So extracellular vesicles are a good platform for developing diagnostic and prognostic tests because in part their abundance and their stability. They're readily found in the blood and they're highly stable, which can't be said for some of these other platforms such as nucleic acids. So what are these extracellular vesicles? Extracellular vesicles are released from pretty much every single cell within your body. So it's not that just cancer cells release them. Most of the cells within your body will release extracellular vesicles. And what you can see is that they are composed of different groups. So we have small exosomes, which are released through the exocytotic pathway, through the multivesicular body. We also have pinching off from the plasma membrane, and these are our microvesicles. And then we have large vesicles as well, termed oncosomes, and also apoptotic bodies. So all of these small vesicles are termed extracellular vesicles. People have tried to characterize an exosome from a microvesicle, but there's been a lot of difficulty in this field. And we know now that a lot of the tetraspanins that were classically considered an exosome or a microvesicle can now be found across multiple ones. So it's really hard to say whether or not something's an exosome or a microvesicle, so we tend just to call them extracellular vesicles. Now, the great thing about the extracellular vesicle as well in terms of developing some type of a liquid biopsy platform is that they are representative in part of the cell of origin. So they contain the tetraspanins, they contain adhesion proteins, and they contain lipids and intracellular proteins and DNAs or nucleic acids that are representative of the cell of origin. So it can tell you something about where it came from, and it can tell you something about the health of that cell. Now, extracellular vesicles in cancer, as the primary tumor cell has been known, we know that it sheds lots of exosomes. You can pick these up in the blood vessel. We know that it could possibly have a role in pre-metastatic niche formation, micrometastases, and metastatic colonization. The important thing here is that all along this way, extracellular vesicles are being constantly shed into the blood. So you can imagine it's a wealth of information sitting in the blood that if you can pick it up and if you can detect it accurately, it can really help guide diagnostics and treatment plans. So the question becomes, how do we study these? And this is where it becomes complicated because we're trying to look at extracellular vesicles in complex fluids. And so a lot of these isolation methods can be very laborious. And this is a good review paper here. This is directly from the review paper showing the most common types of isolation methods that are used and the amount of time that it takes for their use. So if we're talking about something that we want to put into a clinical setting, you can't very well have someone doing differential centrifugation on extracellular vesicles to try and then look at biomarker composition within them. That's not quite realistic. And so these are commonly used to look at, to isolate all extracellular vesicles and then to look at the different components that are in them. There's disadvantages and advantages to each method. It's nicely highlighted in this review. A lot of, probably one of the big disadvantages is that when you're doing a lot of these high-speed spinnings, you're pulling down your extracellular vesicles, but you're also pulling down aggregates and other things that are going to be bound there. So you're not really getting a pure sample, and you might be detecting proteins and so forth that are actually not associated with the extracellular vesicles. Now, the downstream analysis methods for these then is, so once you've isolated your extracellular vesicles, then you can put them to these different types of methodologies. Some of the common ones that are used are digital PCR, then there's omics technology. So a lot of times people have done studies where they've isolated extracellular vesicles and then they've looked at the different RNA, DNA components that are in there, microRNAs as well. And so those can be useful. And then there's Western blot and ELISA as well. Now, the same type of thing falls here that to get to the point of doing analysis, you have to do these very laborious isolation methods, and that's not really ideal in a clinical setting. The other problem that we have with these isolation methods is that you're pooling together all the extracellular vesicles. So if you have this small population that has a unique signature, you're not going to see that. You're not going to pull that out. And so that's a problem as well, too. So one way that we can overcome this then is by using NanoFlow cytometry. And I'll just highlight a couple good reviews here that look at, that are important that look at important considerations for if you're going to do nanoscale flow cytometry, to look at the different parameters and to look at the different effects that everything from your buffers to your settings are going to play a role in how you're going to be analyzing your population of interest. But the important thing about the NanoFlow cytometry is that it represents a really powerful tool for actually being able to look at single extracellular events, and to be able to look at these single events and to be able to analyze them and to be able to look at specific markers on them. And so for this in particular, if you want to know if someone has prostate cancer, a great way to be able to do that is to be able to look at EVs from the prostate. So if you can do some type of phenotype, if you can say this EV came from the prostate, you can then start looking at what are the components that are on that EV or within that EV that might tell you something about whether or not it's a healthy prostate or a diseased prostate. And from that, then you can then develop what would be a multiparametric analysis, where you could look at multiple different markers on your vesicles, and you could say whether or not that extracellular vesicle that came from the prostate happens to have a marker that's associated with cancer or not. So that's some of the power that we see with the ability to look at single extracellular vesicle analysis. Now, this is what we run in my lab, is we run the Saito Flex Flow cytometer. And once again, the purpose is that we want to analyze and quantitate extracellular vesicles in complex biofluids without isolation. Now, for this method, we use the violet light, and we use that to increase light scattering, which increases the resolution of smaller particles. So this is the idea where we really want to be able to push the detection limits to be able to see how small can we see. And so we change our thresholds and we change our triggering so that we can get in between the noise and when we start to actually see some of our particles come up there. A couple things about our method. So we haven't published this, but we'll be putting together, we're working on this paper in collaboration with Beckman to try and put this technology out to really show how we go about using this system to analyze extracellular vesicles in a highly rigorous way. And so for most of the things that I'm going to show you, just a couple of the methods that we use within all of these is that we always use pre-conjugated antibodies. This is important because we don't want to do washes. As soon as you start doing spins and washes, you start losing some of your sample. And then we also dilute our sample in PBS, in a 0.02 micron filtered PBS, and that's how we run it. It decreases our background noise. And we also run on slow. This is really important because standard flow cytometers, if you run them on fast. If you run EVs on fast, you will not see anything. So you have to run it on slow. And we store, we run in milliQ water. We have found that milliQ water has a much, has less noise in it than sheath. But we do store in sheath because sheath has the properties in it to prevent any type of growth within your lines. And then one of the biggest things, we've heard this from other people talking over the last day, is to really do rigorous cleaning, especially when we're talking about extracellular vesicles. So when we're talking about EVs, if you don't do a high level of rigorous cleaning of your machine, you're going to start getting a lot of background noise. And then the other consideration is that we always look at is consider the fluids that everything is in. If your antibody is in glycerol, make sure your control antibody is in glycerol, because if it's not, you're going to see different populations show up because of different refractive indexes, just like if you're going to run something in sucrose or not sucrose, those types of things. And something else to note that we always have is a time count plot. And this is really important when you're running small particles, because when you're running small particles, you really want to see if anything's getting stuck or if there's any type of a clog. And a time count plot is going to show you this. So those are just a few of the things that we always make sure that we have set up so that when we run, that our analysis looks really nice and that we're really confident in what we're seeing in our populations of interest. So what can we do? How far can we push the detection limits when we're talking about looking at very small particles? We know that extracellular vesicles can range between 30 nanometers up to a micron. So what are the parameters that we look at, or how do we look at these small particles on the Saito Flex? So here you can see what we're looking at here is 80 nanometer and 100 nanometer silica beads. So obviously we run silica, not polystyrene, because of the refractive index. And these ones are, we also have the latex ones are fluorescence here at 80. So these are the apogee beads that we're running here. And we can nicely see populations showing up for our 80 nanometer and for our 100 nanometer. When we do our other beads, our fluorescent 100 nanometer show up about there. And then as we start getting bigger in the 240 non-fluorescent, and then the 590 non-fluorescent. And so we get these nice populations that show up with how we have our settings for our thresholding and our triggering on our violet side scatter. Now, when we're running these, just to show some background noise here, as I said, we run in millicu water rather than sheath, and you can kind of see that here. If you run in sheath, you'll see that you have a log 2 over here, whether if you run in millicu water, it's just a regular log 1. And so this is 0.2 micron filtered PBS in sheath, and this is 0.2 micron filtered PBS run in millicu water, and you can see that you get less background noise coming through in the millicu water. Now, obviously our clinical samples that we have are plasma. So how does plasma look when we run with our 0.2 micron filtered PBS? And here's how plasma looks. So this is our plasma diluted 1 in 30 down here. So you can see it here. So we have this large population of stuff. And I say stuff because you can't really tell what it is from here. So then the question becomes, what is this? What portion of this is extracellular vesicles and what portion of it is maybe protein aggregates? And so to start answering this, we did size exclusion chromatography to start. And what is here is the Eyes on Science column. This is considered the gold standard for isolation of EVs with size exclusion chromatography. And what this does is it allows you to isolate your extracellular vesicle fractions. And so when we ran our plasma through this column to isolate our extracellular vesicles, then we're able to run our pure extracellular vesicles and see where they show up on the flow. Now, to do that, we ran it with tetraspanin for CD9. So the CD9 marker is considered to be found on both small exosomes as well as on microvesicles as well. So we use this marker then to look at, okay, so within our purified EVs, so we know these are EVs we're looking at, what portion can we see on flow cytometry when we look at adding in some antibody there to also show that these are extracellular vesicles. And you can see here that this is in our first fraction that we collect, the larger EVs, and here's where they run on the flow. And once again, remember these are beads that we calibrated with beads, so the refractive index of these might be slightly different. Now, when we run the second fraction, where we would expect our smaller EVs to show up, what we see here is that we see a nice population showing CD9 positive events, and we see a shift. So you can see here we have a lot showing up between 100 and 200, and here there's a bit of a shift where we're getting a lot smaller, and we're having quite a few that are showing up lower than the 80 nanometer. Of interest, if you look at just the total population down here, you'll also see that this is our first fraction, and you can see a distinct shift when you go to this second fraction, suggesting that our EVs that are not labeled, you can detect down here. Now, of course, we need more than this to say that they're EVs, so we did some electron microscopy of it. So here's some EM. Here's fraction 1 for the EM. You can see here's our nice extracellular vesicles here, and these ones are running probably around, most of them are greater than about 100 nanometers. There's also some smaller ones in our first fraction too. Now, if we do EM on our second fraction, you can then start to see these much smaller vesicles showing up here. So you can see all these nice lipid structures in the EM, and it gives you an idea of what is the size that we have in each of these fractions that we've isolated. Now, if we do nanosite, so the nanoparticle tracking analysis on this, then you can also see a similar thing, where if we look in our first fraction, you can see that a large portion of the EVs run greater than 100 nanometers in our first fraction. And then in our second fraction, where we would expect our smaller EVs to be, you can then see a shift, where we have a lot that are smaller than 100 nanometers. So the nice thing, what looks nice is when you line all this up together and you say, okay, what are the flow capabilities when we compare it to kind of standard techniques for characterizing or identifying, saying that these are truly EVs that we're looking at. And so you can really see that here, where here's our population, what we say are larger EVs. They're also representative of larger EVs over here, around 100 to 200 nanometers. And then we see the same thing down here with our nanoparticle tracking analysis. And then over here as well, you can see the same things line up, where we see a lot more smaller EVs, and they line up down here, down to about 30 nanometers, suggesting we might be seeing some very small events here with the Saito Flex. And then this lines up down here too, with saying that you can see that a large portion of them run into that smaller range. So all these parameters looked great. We also did some isolation and EM just to see how do those later fractions look when you're getting things a little bit messier, when you're getting some protein aggregates in there. And so this is just all the fractions on EM and how they look on flow, once again with the CD9 marker. And so you can see as you move across a similar trend where we've got a larger, we're shifting over, and then as we start to get our protein aggregates over here, we see a loss in CD9. We also see these tails that come up, and we often see these, and we attribute them to likely that we're looking at protein aggregates. And so it's just a nice way to be able to see how your different fractions run. So now these are on isolated fractions. Obviously, for a clinic, you don't want to be doing any type of the platform beforehand, where you're doing any type of ultracentrifugation or any type of isolation. You want to just be able to run plasma straight. So if we then run our plasma straight with our CD9 antibody, how does that look? So once again, it's our standard where we have 10 microliters of plasma. We incubate with our antibody. There's no washes or anything. And then we dilute in PBS, and then we run on slow, and we collect data for 30 seconds. And this is how it looks. And so this is really nice and kind of representative of if you were going to pool all those fractions together, that you can really see this nice population of CD9 positive extracellular events. Now, when we ran whole plasma, what you also see is this larger population here. And so we're wondering what that larger population was. If we look at our sizing, this is probably around our 500 nanometer. These are probably quite large, so we figured let's pellet them out and take a look. So the postdoctoral fellow in my lab, Dr. Selman, she did some EM on them, and they looked like platelets. So using a platelet marker, we could very quickly and rapidly see that these are indeed platelets. And so you can see that here nicely. There's our platelet. What we've done here is we've gated on this and said how many of these CD9 positive events are also positive for CD41. And there's a nice platelet population showing up there. And this is kind of standard and expected to see because in Canada anyways, most of the biobanks, when they spin down the plasma, they spin it at a speed and a time that what you'd end up with is more platelet low plasma, but not poor. So it wasn't surprising that we were seeing some platelets in there. Now, you can, if you wanted to, you can eliminate those platelets simply by doing a filtration. So this is Postfilter CD9 analysis. And this, of course, is just to obviously show that we run isotype controls for everything. Something that's important here, too, when it comes to running controls is not only do we run individual isotype controls for each antibody, but we also like to run our actual antibody of interest with the isotype control for the other antibody, just to see if there's any interaction or any aggregation between those as well. Now, just to go even further with validating these extracellular vesicle events, we then just did a detergent treatment. So this is a standard way of saying whether or not something is an extracellular vesicle, is that it should be detergent sensitive. So if we add Triton to these, we lose our populations. And that's exactly what you see here. So this population that was there is no longer there once or fairly significantly reduced once we add the detergent treatment. So now just a couple comments to make about this for people who are looking at doing extracellular vesicles platforms. One of the things that we look at doing is when we have an antibody and we have it working, we do some optimization of the antibody to make sure that we don't have excess. Because for all of this work, when we're looking at small particles, the last thing you want to be doing is adding in anything into your sample that you don't need. So an excess of antibody is not something that you ever want to have in your sample. So one of the things that we always do is that we do some antibody titration, where we make sure that we optimize where's the state where the amount of antibody that we have gives us our maximum amount of population rather than when you could add too much antibody and then you've just got excess antibody in your sample, which once again, when you're doing things on a small scale like this, you want to try and eliminate that. The other thing that we look for as well is we look for swarm or coincidence incidents, just to make sure that we are truly measuring single particle events. And this is simple to do, you just dilute down. So here you can see our CD9, we have about 8,000 events. When we do a dilution series, we can see that that's dropping. We have about 4,000 and then 2,000. So it's a very quick way to show you whether or not you have more than one particle in the stream, which, as you can imagine, is an issue if you have a lot of EVs. And once again, that's where you set your parameters for where you're going to dilute your sample. We dilute our plasma 1 in 30. We find that we do this and we don't get any coincidence analysis. So now, how does this play out when you actually want to take this to the next step? So I think a lot of this really shows the utility of NanoFlow to be able to identify extracellular vesicles. And so now what do we do if we want to say, okay, we want to know whether or not someone has a status of health or if they have a status of disease. So this is just a bit of a story that we've been working on that we're putting together right now. It's not published yet, but we're aiming to do that soon. And so this is looking at prostate cancer. And so this deals with, as I kind of mentioned, one of the overtreatment issues that we have in prostate cancer, where most men are screened with the PSA test when they turn 50 or around 50 anyways. And what we know with PSA testing, this is from the NIH and the Canadian guidelines, is that for every 1,000 men that are screened, PSA is going to be elevated in about 230 of these men. And from these 230 men, they will all undergo needle core biopsies. What we know is that half of them don't have prostate cancer at all. They either have a benign disease or it was just a misdiagnosis. And so that's represented here as those people there. Now, we also know that we have 40% that will have prostate cancer. So this is what we call our non-indolent form down here. Rather, these people have an indolent disease that you don't really want to detect because as we age, we all end up with small growths within our body. Whether or not you want someone to find them is not likely, especially when it comes to prostate cancer. And then we still notice that there's about 3% of prostate cancer cases that are missed. So we looked at whether or not we could use NanoFlow to develop a test to say, if we can characterize EVs that have come from the prostate, can we then look at markers that are on them to say whether or not that has come from an aggressive prostate cancer? And so just for a little bit of background about some of the scoring in prostate cancer, so prostate cancer undergoes a classical scoring system, Gleason scoring system, and the Gleason scoring system decides whether or not someone is low risk or if they have high risk aggressive disease. So here you can see someone who's low risk, they have well differentiated glands, and as you move more to a dedifferentiated state, so you can see a lack of any type of a glandular formation there. And what we know, so a lot of the, whether or not we're calling someone low risk or high risk, comes from their Gleason score. And this is because we know from long-term studies that if someone has a low Gleason score, they have a very good long-term outcome and likely are not in need of treatment. And that is shown here by this study that did a 20-year outcome following conservative management of clinically localized prostate cancer, where they showed that individuals here, so this is our low-risk group here, in the dark line is deaths from prostate cancer, in the light gray is death from any other possible reason why an individual might die. And as you can see, for these people with the low-grade group 1 prostate cancer, their risk of dying from their cancer is much lower than their risk of dying from any other event. But then once we start to get to these higher groups, all of a sudden you start to see that then that changes. You can see here for this group in particular, their risk of dying from cancer is very significant. So what we wanted to see was whether or not we could develop a liquid biopsy that could say, okay, we want to be able to detect these people down here, but we don't want to detect and treat these people here. And so this work was started with Dr. Han Liang when he was at Western, and it was looking at a couple prostate-specific markers. So this is once again important because you're picking up a lot of things in the blood. There's a lot of extracellular vesicles in there. You want to have some type of a tissue to look at. So you want to know where your extracellular vesicle has come from, and that's where our platform is, that we start by looking at a tissue-specific marker. So to look at whether or not something is coming from the prostate, PSMA was one that has been worked on quite heavily. It's been worked on by a few groups in Canada in terms of extracellular vesicle liquid biopsy development. And while it is elevated on the prostate, and it is called prostate-specific membrane antigen, it's not solely specific to the prostate, so it is found in other places. So we did a combination here where we added in another marker as well, which is STEAP1. This one is definitely specific for the prostate. And so the idea here was that if we can find these on EVs, we can say that these are EVs from the prostate, with the plan of saying that we can do this in a flow cytometry-based method. Now, if we have the markers to say this is coming from a certain tissue, which we have with the STEAP1 and the PSMA, how do we know whether or not that tissue is healthy or diseased? Because that's always one of the big questions. And for this, you need other biomarkers that come into play here. So there's ways that you could go about either trying to find new biomarkers, or if there's biomarkers of interest, as was kind of talked about in this morning's talk, where people flock to whatever is new and going on in literature right now, then sometimes that's a good place to start. So this was one of the markers that I discovered during my postdoctoral fellowship that is on prostate cancer. Once again, none of this has been published yet. We're in the process of putting this out. So what I found was that polysialic acid is present on prostate, on aggressive prostate cancer cells. Polysialic acid is a very interesting glycan. It's not a protein, it's a sugar. And I have a particular interest in sugars in cancer biology. And so this sugar is a long chain of alpha-268 linked sialic acids. And having these sugars present on a protein dramatically changes how it interacts with its environment and how the protein that is carrying this glycan also functions. And so it's been noted in a few other cancers before, such as neuroblastoma, glioblastoma, and small cell lung cancer. The expression of polysialic acid is pretty much restricted to the nervous system. It's important in development during neural crest cell migration and so forth, but otherwise, outside the neural system, sometimes immune cells express it as well. It tends to be fairly localized to specific cells. So seeing it on prostate cancer was very interesting and suggested the possibility that this might be a biomarker that we could add in to our extracellular vesicle test to try and get better risk stratification in prostate cancer. And now we had just using basic cell culture methods, we had some evidence that the polysialic acid that was expressed on these aggressive prostate cancer cells was being released in vesicles. But before we jump too far ahead, we wanted to make sure that our biomarker that we wanted to look at in the blood, being released from prostate cells, was actually present on prostate tumor tissue. And so for this, in collaboration with Dr. Michael Cox here at the Vancouver Prostate Center, we did a lot of staining of, well, I shouldn't say we did, Mike did a lot of staining of prostate cores from individuals that had long-term outcomes. And so this is important because, once again, we know that with prostate cancer, we want people that are going to have long-term poor prognostic outcomes. So with this tissue microarray, we were able to say what are the levels of the expression of this carbohydrate in an individual's tissue, and what was their outcome 10 years post-diagnosis. So the results from this, as you can see here, so here's some histology staining where we have a benign condition. These are the low-risk group 1, and then as we increase across our groups, you can see an increase in the staining of polysialic acid. We then could correlate this with patient outcomes, and we could see that a moderate to strong level led to a significantly worse outcome. And this also correlated with the number of patient deaths that you could see here. So all of this supported that, yes, polysialic acid could be a new and interesting biomarker in prostate cancer. So then we went to look at it on extracellular vesicles. So can we find it on extracellular vesicles? And here you can see, using flow, that we've got this really nice population of polysialic acid on the flow. Now, the nice thing about studying carbohydrates is often they have enzymes. Well, in this particular case, bacterial phage creates this enzyme, and it selectively cleaves off polycylic acid. So this gives us a unique control. What we can do here is we can take our plasma, we can add the enzyme that should cleave all of the polycylic acid off, and then we add in the antibody that detects polycylic acid. And what you see when you add that enzyme is you see a loss of your signal. And this shows that this is specific to that carbohydrate. And then when we did an analysis of many of them, and you can see that you see a significant reduction, suggesting that this is truly polycylic acid, and it's on the extracellular vesicles, and we can detect it in plasma. Now, we also wanted to validate this further, because once again, just seeing things on plots is also nice to do other validations using other types of techniques that are recommended. And so for this, we did, once again, size exclusion, and we did EV isolation, and then we did Western blot. Now, the markers that we used here are your standard markers for looking at EVs. If anyone wants to look at any more markers than this, there's the MISIV guidelines, so the minimal information for the study of extracellular vesicles, which is published in 2018. It has a whole list of different markers that you can use to validate that what you're seeing is truly an extracellular vesicle. So we ran our fractions, and you can see here TSG 101, CD63, and CD81 all associate with what we'd expect to see as vesicular fractions. And then within that, we see that indeed our carbohydrate polycylic acid is present on these extracellular vesicles, once again validating that it's truly a vesicular format that we're looking at and seeing here. Now, when we looked at it in patient samples, polycylic acid on its own didn't really tell us too much. And once again, I think this really highlights one of the things that you can do with NanoFlow that you can't necessarily do with other methods is you can bring in other markers. And so we then looked at whether or not it was actually on a extracellular vesicle from the prostate. Because if it's not on an extracellular vesicle from the prostate, it could be from an immune cell, it could be coming from somewhere else. So looking at just a single biomarker and its levels doesn't necessarily tell you as much as if you can say, what is that biomarker level on that tissue? And we use that by looking at the EVs that are coming from that tissue. So when we did that, here's our fraction on our flows. When we did that, we could see that polycylic acid was clearly present on these extracellular vesicles that were coming from the prostate. And once again, these are just our fractions, our EV fractions, and then fraction 4, where we'd start to see some more protein contamination. Within each fraction, you can see the levels of STEAP1, polycia, and PSMA. And then what we can do here is we can take our STEAP1 positive extracellular vesicles, and we can gate and say how many of those are also positive for PSMA and how many of those are also positive for polycia. And then that gives us our population here. So we can see our triple positive population here, we can see our dual positive population down there, and then we can see ones that STEAP1 extracellular vesicles that are not positive for any of those markers here. And so within our triple positive, you can see that these nicely line up, suggesting that all of these can be found on the same vesicular event. And just to note, you can once again see a bit of a shift too. When we look at our fraction 2, which would possibly be slightly larger extracellular vesicles, versus 3, you can kind of see a bit of a shift to a smaller size there, which is also nice to see. So how does this look when we actually run all this together in a cohort? So we've got our marker that says that this extracellular vesicle has come from the prostate. Now we've got our cancer marker that says that this possibly means that someone has an aggressive disease. So what we did was we collected a cohort. This is just a small cohort of 87 men. All these men were diagnosed with using PSA as having prostate cancer. And so they all underwent a needle core biopsy. And that needle core biopsy gives us that Gleason score. And that Gleason score is what allows us to say whether or not they're low risk or whether or not they're high risk. What we obviously want to do is we want a test that's going to make it so that people don't even have to go in for that needle core biopsy. We want them to be able to skip that and to be comfortable knowing that they have a low-risk disease. So all the plasma was collected, and the important thing was the collection. This is another thing that to take into consideration when you're doing EV work and EV analysis is the type of collection tubes that you're getting from biobanks. Biobanks will use different collection tubes. There's also recommended collection tubes that you'll find in the MISAV guidelines as well. EDTA is a great tube. In my opinion, it's also one of the ones that's recommended. And it's a common one for certain biobanks, but not all do it. Some do citrate, some do heparin. But it's important to know, because if you know and report on what the tube is, then that lets people know how your analysis is looking and how the tube that was collected in might be impacting what you're seeing. All of these plasma samples were collected in the exact same way. They were spun in the same centrifuge, in the same rotor for the same amount of time. So everything here was done identically. And we even have time to collection, so we know how long it took for them to take the plasma, for them to take the blood, do the spin, and then put it in the liquid nitrogen. And that was all done within 2 hours. So that's kind of the best possible cohort that we could set up to say whether or not we could then use what we were looking at on the flow by detecting these small extracellular vesicles and see what these levels look like in low risk and high risk patients. And here's just some of the flow data from that. When we look at a low-risk individual, and this one's just, there's obviously a lot of heterogeneity in how these samples look. This one I'm showing just because it's, once again, just the importance of being able to look at more than one thing. If you were going to do something on an ELISA of extracellular vesicles, an ELISA of extracellular vesicles wouldn't tell you about the composition of the biomarkers on that vesicle. NanoFlow can tell you about the composition of the biomarkers on that. And that is, you can really see here, because this person has really high levels of STEAP1, but also really high levels of polycia. But when you look at the dual positive population, it's not that high. When you look at this person who's high risk, what you can see is that they have a very high dual positive population. And this just is important because depending on what an individual is doing, maybe this person went biking the day before and they happen to have more STEAP1 positive EVs in their blood. Maybe they are fighting off a bit of a cold or have a bit of an infection, and that's why their polycia is elevated. Those could be any number of reasons for why we're seeing high levels of an individual extracellular vesicle in the plasma. But once we start being able to look at things in dual and triple positives, then we start to be able to get a much better representation of someone's health or disease status there. Now, we also looked at PSMA, and we then looked at the triple positives too. And so this is the plots that we show here, where here we ask how many of our, so this one's gated on P4, how many of our STEAP1 positive events are also positive for polycia and for PSMA. And so this is what we use here. So our readout is we run for 30 seconds, we collect the events, and then the events is what we display. So we say how many events are in here, and then we do a comparison of how many events do low-risk people have, how many events do high-risk people have. And then just to note, we also did look just at a classical extracellular vesicle marker on these, just to show once again that these are indeed vesicles. You can see that here with CD9, that you can see that a lot of these triple positive events also localize with a CD9 marker on them as well. Once again, suggesting that they are extracellular vesicles that have been released from the prostate. So how does it all play out when we do the analysis of the data from this and we actually look at the number of events that we are seeing in low risk and high risk patients? So once again, those low risk, the Gleason score 1 and Gleason group 2, which is high risk, but some people in this risk group do progress, but there's no good way to determine which people are low risk and high risk within this risk group, so that gets a little complicated. And then we have our high risk here. When we put them together, you can see low risk and high risk. So here you can start to see this population here almost separates out from the others. And this gives you your triple positive number of events that we're seeing there. And we can see that there's definite statistical significance in an increase in the number of triple positive extracellular events as detected by NanoFlow. Now, we did notice that we do have a few people down here that were not detected. And so we thought, can they be detected through PSA screening? So can we do a combination here? Can we look at an individual's PSA levels, and then can we also look at a triple positive EV test to see if we can improve our ability to risk stratify these patients? So we looked at people's PSA levels, and then we looked at their triple positive levels. And what we know is that for a lot of people, clinicians, when you have a PSA level that is usually under 15 or under 10, particularly under 10, that's often when clinicians don't really know whether or not you have an aggressive disease. And so a lot of these people get sent for biopsies because they're really not sure with that type of a level. When you start going higher than that, when you have someone who has a PSA of 18 or a PSA of 20, they have a 99% chance of having prostate cancer. So in that regard, the PSA is very sensitive when it gets up that high. And so you can see here that if we look at the level of PSA in the relationship to the level of our total EVs, that we can really start to kind of group people. So we can say, here's our low-risk people as determined by their triple positive levels. And up here, where we have either high PSA or high triple positive, these people are high risk. So you can kind of start to see that grouping there. And then we asked, what are the test parameters of this? So if we wanted to look at the sensitivity and the specificity of this, where does this fall? And what we can see is when we look at PSA and triple positive together, that we can get an AUC of about 0.827, which is pretty good in terms of test development. One other thing that pretty much wraps up that story, but I just did want to highlight one other thing about, can you run your sample on a different day with a different antibody, with a different conjugation kit, and still get the same results? And you can. So here is on a run that was done on one particular day, and here was a run after we had run out of antibody and the student relabeled some and ran it again. And we see very good ability of the Saito Flex to give accurate reads from one day to the next. This does have a lot to do with keeping your machine really clean as well, but we do see really great reproducibility within this, which makes it quite promising as we see. So in summary, hopefully everyone from this can see the power that we see for NanoFlow and the utility of the Saito Flex for this type of a platform. Overall, we know that we don't want to do, for a clinical setting, we want to avoid the laborious methods of doing any type of a purification. We want to just be able to run sample and to be able to do analysis on it. And NanoFlow cytometry really gives us a platform for doing that. Now, the other thing, though, is that with NanoFlow, you do have to have your markers of interest. That is the thing. So if you wanted to do new biomarker discovery, being able to sort EVs would really be the way to go. And in very good timing with this week is this paper just came out. And I'm really excited about this. They did some sorting with the MoFlow, the Beckman MoFlow, and they showed that they could sort EVs. And it was a great paper that came out. It's been something that we've been wanting to do in the lab to say, can we do some new biomarker discovery? And can we look at different populations of EVs depending on their biomarker expression? So that's some really exciting work that just came out from Dr. Jones. So there's, once again, in summary, we really think that NanoFlow has a lot of potential to be able to be used as a diagnostic and prognostic and to improve how we detect cancer and how we do patient screening. And then hopefully the testing that I showed you there really shows you the power when we're looking at improving our ability to prognosticate prostate cancer patients. So then just some acknowledgements for that. So there's people in my lab. The main bulk of this work was done by Dr. Salmond, a postdoctoral fellow in the lab, and a PhD student, Karin Kana. Collaborators, Dr. Leong, Dr. Mike Koch, pathologists, and urologists who are all responsible in clinical sample acquisition. And then over at Beckman as well, in particular Darren at Beckman, who really helped when I was getting this platform up and running and dealing with my millions of emails on how to get the Saito Flex set up and just playing with these types of things, getting beads on there, getting the system running in such a way that I was really happy with how the data was being reported. And then funding as well. And so that's it. Thank you.